Welcome to the Jackson Laboratories Humanized Mice PubCast, where we offer a glimpse into the world of humanized mice through the window of a recent scholarly publication. In this series, we're defining humanized mice as immunodeficient mice that have been engrafted with human CD34 positive hematopoietic stem cells or a human PBMC-derived humanized immune system. JAX is a longtime leading provider of both naive immunodeficient and humanized mice to the life sciences community worldwide. We regularly run preclinical studies for external clients using these models, most commonly in the fields of oncology, immuno-oncology, and transplantation biology and drug development. But we're always excited to see the new and different ways that other researchers use these models. Join us as we explore the cutting-edge research and applications of humanized mice.
Welcome to JAX's Humanized Mouse PubCast. I'm Charu Gupta, a technical information scientist, and today we will discuss a recent study from Penn State's Zen Lab published in the Journal of Immunology, titled Acute Myeloid Leukemia Causes T-cell Exhaustion and Depletion in a Humanized Graft-versus-Leukemia Model. In this article, the authors sought to develop a more clinically relevant model to study and target acute myeloid leukemia, as well as its interactions with the human immune system. Now, before we delve into why there was a need to establish a model that better recapitulated the human disease, let's briefly review AML. AML is a type of cancer that affects the white blood cells in the blood and the bone marrow. It is characterized by the acute or rapid proliferation of immature white blood cells, or myeloblasts. These are abnormal cells that remain in an immature state and are unable to function properly and can build up and crowd out healthy white blood cells, red blood cells, platelets, etc. Compared to a normal blood population, the AML sample is primarily composed of immature progenitor-like blood cells, or myeloblasts. Blood cancers are the fifth most common cancer worldwide, with 32% being leukemias. Symptoms may include fever, bone pain, lethargy, shortness of breath, and easy bruising. The progression of AML is rapid, and this can be fatal within weeks or months if left untreated, highlighting the importance of needing reliable models to study and develop more efficacious treatments for the disease. Now, standard of care options include chemotherapy or targeted therapies such as molecular inhibitors and antibodies. Stem cell transplants, or SCTs, are considered for patients who are at high risk of relapse or have relapse, and it is the only curative therapy. Now, these transplants can be autologous, which means that the cells come from the patient themselves, or allogenic, which means that the cells come from a donor. This donor could be a blood relative or unrelated, as long as their cells are a close match to that of the patient. Now, allogenic stem cell transplants are the most common types of SCTs used to treat AML, and these stem cell transplants will work against the cancer directly. This happens because of an effect known as graft versus tumor effect, and in this case, graft versus leukemia. The graft versus leukemia reaction refers to the ability of a donor's immune system to eliminate host leukemic cells after allogenic HSCT. GVL can help reduce the relapse of leukemia. However, the success of this therapy is only 50%, because half of the patients relapse after 6 months of receiving this treatment. This is due to several reasons, which include and are not limited to hidden residual leukemia cells, cytogenetic and genetic factors, as well as immune evasion. Now, to enhance the curative effects of this treatment, it is necessary to truly understand why some stem cell grafts lead to the relapse of leukemia while some are accepted. And therefore, the authors embarked on a journey to create a preclinical model that better recapitulates the graft versus leukemia observed in the clinic, hoping to gain translational, mechanistic, and therapeutic insights. The naive model, or the mouse model prior to engraftment that the authors used, is the JAX NSG-MHC class 1 and class 2 double knockout mouse, which is stock number 25216. Now, because the MHC class 1 and class 2 double knockout mice have a delayed graft versus host disease, the experimental window is much longer, allowing for immuno-oncological as well as immune activating studies, which by nature are a little bit longer than a couple of weeks. With the aim being to establish a clinically relevant graft versus leukemia or GVL model, the authors began by sublethally irradiating the MHC double knockout NSG mice, and they were then intramurally engrafted with the AML. The irradiation helps the AML engraft better. 4 weeks later, once the AML was established, the mice were immune humanized with donor immune cells. These cells were obtained from the AML patient 6 months post stem cell transplant when they were in complete remission. So we can say that 99% of these PBMCs belong to the donor. 4 to 12 weeks later, a mouse model was created that would replicate the inpatient condition post stem cell transplant. Now, to characterize and validate this model, the authors looked for the first hallmark of GVL, which is slowed down AML kinetics due to the newly engrafted healthy immune cells, in comparison to the AML engrafted but non-humanized mouse. Now, the authors compared AML growth kinetics by measuring CD33 positive circulating cells from the peripheral blood over 12 weeks from AML engrafted NSG-MHC DKO mice with and without immune cell engraftment. The graph on the right notes that the AML-only mice have a faster growth curve as compared with the immune cell engrafted humanized AML mice. We therefore have observed slowed down AML growth kinetics due to immune cell interference. Let's take a look at the immune cell subsets that are causing this hindrance. Now, immune cell data from mice engrafted with PBMC alone from 3 different patients is presented here from the blood, bone marrow, and the spleen. While T cell, B cell, NK, and myeloid populations, monocyte populations are noted, the authors observed the highest population to be contributed via T cells. This is very normal for a PBMC humanized model, as the mouse just does not have the transgenes to support NK or myeloid engraftment. The T cells are able to expand, and this is the population that was studied further. The next hallmark of GVL would be immune cell dysfunction and depletion. Now, due to their large population, the authors concentrated on observing T cell counts in mice engrafted with PBMC only versus PBMC plus the AML or the GVL model. Now, looking at CD3 positive T cells in the blood, bone marrow, and spleens of the engrafted mice, we see a decreased cell count across all 3 tissues in GVL mouse, and this reduction in count was owed to increased cell apoptosis. A marked decrease in T cell cytokine release with heavily reduced effector cytokine IFN gamma was noted, and a lowered cytotoxicity was also noted due to the now lowered levels of granzyme B. These point to T cell exhaustion. The final validation of the GVL model resides in its ability to prove itself as a translationally relevant therapeutic model. The authors decided to run a proof of concept study to remedy the T cell exhaustion in the GVL model. Now, AML engrafted MHC DKO mice were co-engrafted with matching donor PBMCs and then treated with monoclonal antibodies against those cell surface receptors of TIM3, TIGIT, PD1 in a combination or their isotype controls twice a week. Now, the first flow cytometric plot here shows the treatment with the antibody combination led to a decrease in the number of CD33 positive circulating cells. These CD33 positive cells are leukemic. Now, next, to address T cell function, CD8 positive cytotoxic T cells were queried for granzyme B, which was increased upon the treatment, indicating regained cellular cytotoxic function, And effector cytokine release was also regained as judged by the increase in the IFN gamma levels. Overall, the triple monoclonal antibody treatment that blocks T cell exhaustion mediating cell surface receptors was able to aid donor immune cells in reducing the leukemia. This certainly proves the usefulness of this breakthrough model in preclinical applications. Now, our story began with a hunt for a translational model for graft-versus-leukemia, where one could understand why half the patients reject the curative stem cell transplant treatment. The established GVL model demonstrates hallmarks of clinical condition with lowered AML growth kinetics due to the immune cells, as well as immune cell dysfunction and depletion due to the AML. This model can be further used to identify markers for transplant acceptance and rejection, as well as testing new therapies. The take-home message here is that a novel breakthrough preclinical mouse model that is able to engraft the AML as well as human immune cells at the same time was created so that we can study the interactions between these two systems outside of the patient body for the first time. This was my brief review of this amazing study. Read it for yourself. Share your thoughts, stay curious, and thank you for joining me today.